Good day everyone, I'm Anjayani as a student of the Faculty of Animal Science, Jambi University. I would like to present our article entitled Use of Merremia peltata to substitute Brachiaria mutica in ruminant diet, an in vitro study. So for the background, the Brachiaria mutica has been the main source of forages for ruminants. Recently, its availability has been limited due to the conversion of grassland to residential and industrial uses. On the other hand, Merremia peltata is abundantly available nowadays and it has a great potential as ruminant feed. It has a highly productivity within the 90 days and can produce 60 grams of fresh wake or 24 grams of dry matter. So the nutrient MP is comparable to BM. The CP and the CF content of MP is higher than of BM, but vice versa for NFE and GE. So for research objectives is to evaluate the Merremia peltata as a substitute for Brachiaria mutica as the main source of fiber in the complete diet of ruminants. So in this table 1, each treatment consisted of 60% of forages and 40% of concentrate. The forage in each treatment was a combination of MP and BM. So in the table 2, as we can see, in general, there was an improvement in the nutrient content of the treatments with the higher level of MP in the ration except for NFE. So in this in vitro study, each treatment was incubated in buffer rumen fluid for 72 hours. The gas production we measure at 2, 4, 6, 8 until 72 hours post-incubation. For the fermentation residue, were recovered through the filter paper and analyzed for ash and used for the calculation of apparently OM degradation. For another fermentation bottle was refluxed with 50 milliliters NDS, and residue was also recovered through filter paper and used for the calculation of truly OM degradation or TOMD. For the calculation for MBO, it's according to the Blamol. For the MN equation, it's according to the De Meyer. And the gas volume according to Orskov and McDonald. So the data were subjected to the ANOVA based on CRD, and the data showing significance between treatment means were separated with orthogonal polynomial contrasts. So the contrasts used were linear, quadratic, and deviated from linear and quadratic. So for the result, the treatment had a very significant effect with P lower than 0.01 on the values of A, B, and C. There is an increasing level of MP in the diet, decreased the gas production of fermentation in the fraction A, but increased the rate and total gas production of fraction B quadratically. So as we can see in this graphic, the P0, the blue one, and the BM as the main fiber source had a similar gas production profile as P2 with 40% MP and 20% BM. And in the P3, with the main fiber supply from MP, had the highest total gas production, whereas the P1 with 20% of MP and 40% of PM had the lowest total gas production. And then the right figure, the P3 P3 with MP as fiber source had the highest fermentation rate, and P0 with BM as fiber source has the lowest, P0. And the additive effect on the fermentation rate was found in the study as shown in P1 and P2 with combined fiber sources of MP and BM. So the treatments had a very significant effect with P lower than 0.05 on TOMD, MBO, and MM, but not on AOMD. The TOMD, MBO, and MM linearly increase with higher level of MP in duration. So the effects of MP level on OM degradation can be seen from this figure 1, and the effects of MP level on microbial biomass production can be seen in figure 2. In conclusions, MP can replace BM up to 73.3% in the ruminant diet with an optimum combination of 44% MP and 16% of BM. For recommendation, the results of this research need to be validated in vivo before they can be widely implemented. For the acknowledgements, the authors would like to thank the University of Jambi for funding this research.